What we'll be talking today is a little bit about using proteins as an example of learning modern science. When people go to high school, there's always the story: you first take biology, and then you take chemistry, and then you take physics. My point is to show that in the modern world, probably we should start to teach high school in the opposite order. And things get integrated. You cannot, when you come to modern biology, you probably should learn physics first, and then chemistry, and then come to biology. So what I'm trying to make a case is how in the modern society, that boring biology course that every high school student try to get out of their front because it's just a sort of descriptions can become something actually very interesting on the present way of doing science. And the beauty of molecular biology is that you look in front of you and say, what are you doing? Biology, physics, chemistry, and there's no separation. And base here is an integration of everything. If you look for which are the molecules that people call macromolecules, these are big biological molecules that most of our body are made from. You have three kinds of them. Some of them are what people call nucleic acids. The other one are proteins we're going to talk about. And then finally, we have carbohydrates, they're sugars. So what you have is DNA. These are the molecules that contain the information. It's like your disk drive with all the information that you need. Proteins are the molecules going to do all the function, all the work on your body, structural, enzymatic, every kind of work that's being done on you, carry oxygen, whatever has to be done on your body is being done by proteins. And the carbohydrates just give you sugars, just give you energy. So they're the boring one. We're not going to talk about fuel. Let's talk about these molecules. The success of molecular biology came from when people figured out that this molecule called DNA, and this molecule contains the information that can be read from the information. On each gene of this molecule is going to have the information to generate one particular molecule they're going to describe to you. There are proteins, there are sequence of peptides. And this information is transcribed into this series of amino acids. We'll be described that in detail. So what you have is the DNA has this information. This information is going to be translated into this big molecule that's like a big necklace where each bead is one amino acid. And this particular sequence of amino acids is going to be able to go to one particular well-defined three-dimensional structures, and each one of them is going to perform all the functions. You have a cellular level at all levels at the organism. So what we'll be describing today is mostly this step. How do I go from this linear sequence, how they can identify their structure. But before we do that, let's describe a little bit about this first step. The first step is really the development of the genetic code that became so powerful. So DNA, if you have heard about, DNA is a double helix, what means you have two chains. So you have two heteropolymers, like the protein. These are also heteropolymers. What do you mean heteropolymer? Like I said, a polymer is just like a necklace, but each bead of this necklace can be different. In the case of the DNA, you have four kinds of bases they call C, G, and A and T. And each of these big chains, they can get together with the other one by pairing some of these groups. So every C get joined with a G, and every A get joined to a T. So what you have is, you have this double helix. Now, how this information from the DNA is translated into a protein. That's where the genetic code appears. So genetic code, if you try to come, is that each three base pairs, if you get one of these chains, each of these three base pairs, each combination of C, G's, A, and T's, get any three letters. Each three letters defines one amino acid. So what you have is, in the cell, it's read. It reads the sequence. These three give one amino acid. These other three gives one amino acid. The other three give another amino acid. So what you have is, you keep collecting and put these beads following each other, right? So you see that you go, and each one of these three ones identify one particular different amino acid, you put this chain. So you read it, just like the way you read a tape drive, you read this information, you put this chain. Your gene is actually a little bit more complicated than that. You're going to have all the other controls, amino acids, they have controls part of it, but most of it, each gene gives you one protein. Right? And this protein is identified, like I said, this is the success of genetic code. That was the first time in biology that people were able to get a very specific effect, how this information is stored in terms of these base pairs. Now, you generate now on each gene this big sequence, this big heteropolymer. So now you have this big protein. So the protein now has this chain of residues, and the question is how this chain goes to make a structure. If you look at this structure, it's a schematic version of a protein. So what I have is local structures. This is when the sequence of amino acids start to form helices, or when they can form strands. So you have the base strands and the alpha helix, and then that's how they organized in a three-dimensional structure. And that's how function is performed. But the amazing thing is that most of the proteins, all they need is, by the time this chain is created, by the time the machine creates this chain, this necklace is able to fold into this unique three-dimensional structure without any external help. So it is a self-assembling mechanism. So the question is, how do we do this translation? So this translation that you're able to go from providing the sequence to get the structure is what people call the protein folding problem, or what some people say, can we find the second genomic code? The code that couples not now this particular heteropolymer, that's a sequence of the DNA into a protein, but how do you convert this sequence into the structure? Why that is important? For several reasons. First of all, people know a lot about this information. Everybody this year has heard about the Genome Project, and we have been able to sequence the entire human genome, and we have the information. What it means is we still have a mass to go over, but in principle, we're going to be able to identify all those sequences. But the question is, if I have those sequences, do I know what the structure is? We don't. So this is the step that goes beyond, but that's what we need for function. Here's cytochrome C peroxidase. So you see it's an example of the folding problem. So what you have is that sequence information, those beads all have names. Each one of them is one amino acid, one of the beads. And the amino acids, they all have the same part of the main chain, but they have different parts hanging on the side. That's what they call the side groups. And that's how they interact. The question we ask now is, how do these things work? Do I need to know all the level of detail of that things? Mostly, people like to say, I need every atom and every detail, every outside. The idea came from physics is that, what's the minimal description that I can have to understand what's the overall mechanism that allows a one-dimensional sequence of beads to fold into a unique structure? Can we understand that mechanism? So the first thing you look at these things is, what characterizes different amino acids? If you look at, there are many, many differences among them. Right, you can see this one, it has an aromatic group that's very non-polar. This one has this oxygen here, this CO group that's very polar. This is just CH, 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 so that's very non-polar. So what means non-polar? Non-polar is just like oil and water. Polar groups like to mix with water. Non-polar groups hate water. So you start to see that non-polar groups like to get close to each other. The polar groups like to get close to each other, expose the solvent, and this is what creates the separation. So what's the basic interaction that defines folding? And people have been working there for some time. So the most important interaction is the one I just told you. Although you have 20 amino acids, they have all these different kinds. The most important interaction that makes amino acids, to make that sequence to try to fold into a unique structure, is what we call the hydrophobic interaction. And the hydrophobic interaction tells you that these groups that are non-polar, that are oil-like, they like to get together. So the dark green is what we call the hydrophobic groups. They like to get together. That's exactly like the way that oil wants to separate itself from water. And the groups that are polar, they don't mind to get exposed to water. So that's the basic phase separation. The entire chain tries to make sure that these polar groups, they all come together to each other. The non-polar try to stay exposed to the water.
And this phase separation creates the structure. So what's the structure? They're going to look for the structure where you can have a maximum of these groups coming together and less of the other groups separate from them. Are there any other interactions? Yes, there are. There are interactions, what we call hydrogen bonds. They respond for making the secondary structure. They're packing interactions, as you'd expect. If you have large groups and a small group, they all have to fit together. So you have to take that into account. There are groups that may be positively charged and negatively charged, and charged don't like to live alone, so that positive charge and negative like to come together. There are things they call disulfide interactions, where sulfur bridges try to come together. But this is the biggest one. And we can describe the main mechanism just thinking in terms of this one. So the question, can we understand the main mechanism of folding? Just think in terms of the simple interaction, hydrophobic interactions and hydrophilic interactions. So let's go next step. So when it came from physics, say, let's step back from proteins. Let's try to define an artificial protein. Or artificial system can help us to describe this system. So the system we thought about it is a system where now, instead of having 20 amino acids as a protein, we are just going to have 2 kinds of them. And to make our life simpler, I'm going to call them A and B. And I'm going to define an interaction that's very simple between these residues. I'm going to say that A likes to stay close to A, B likes to stay close to B, but A doesn't like to stay close to B. That's a very simple interaction. Is that exactly like a protein? No, proteins have a much broader possibility. They have 20 amino acids, they have all these other kind of interactions. But this is enough to say, can I now, by just using 2 kinds of amino acids, create sequences that fold into a unique structure? Can I find what takes for a sequence to fold into a unique structure? This was the basic idea. Can we squeeze that? Understand which kind of sequence are able to find a unique structure and which ones are not. And that was the basic idea of physics coming. That was the basic idea of landscapes trying to show and help us to understand biology. So with that in mind, we're in very good shape now. See what we did. We completely destroyed all the details and tried to keep the essence. Can we, with this essence, understand the mechanism behind driving fold? So you see, that's something that made for a long time a big collision between the physics community working on this problem and the biology community working on this problem. See, here you have a protein. It's all the atoms, it's all the details I showed you before. They still a heteropolymer, but a very detailed heteropolymer with 20 kinds of amino acids with all the details of every atom. And here I'm defining also a heteropolymer. That's a sequence, but instead of being made by 20 kinds of different beads, I'm just using 2 of them, and with a very simple interaction among them. But the question is, again, can we figure out that, in essence, how we can seek for a specific structure doing that? This energy landscape idea, actually, I want to go back to history. The folding problem starts by a person called Amundsen. And Amundsen did a very simple experiment that created this entire field. He did, on the late 50s and early 60s, an experiment that was illegal in biology during that time. And what I mean by illegal in biology is, if you study the system out of their physiological conditions, they are of no relevance. But he did that, and he understood an interesting problem. What he did, he got a particular protein called ribonuclease, and he unfolded that protein. You can unfold the protein by many different ways. That's by denature a protein, and we do that all the time. You can put some acid there, like by adding lime. You can heat it up. You can mix up with urea. Anything you want is going to denature that protein. And then what he saw is, this protein didn't work. But then he put back this protein into physiological conditions. And what he saw, the protein started to work again. So he said, this protein is able to fold alone. You don't need any extra help because they destroyed this protein, doesn't have any of the cellular machinery now, and is able to go back and work. We try to do the experiment at home all the time, doesn't work. Why doesn't it work for us? Basically, you go in the morning and you fry an egg. And most of the egg, you have lots of proteins on the egg white. And you see that after your egg is fried, and you lower the temperature of it, you don't see the egg going back to the original shape. You have the fried egg doesn't come. The reason the fried egg doesn't come is because these proteins are on so large concentration. The concentration of that protein is so extremely large that when you unfold it, what happens is these hydrophobic groups, instead of fold together, they come together. So what you have is the same way oil tries to separate from water, these hydrophobic groups, but this one from one protein gets to the other one and gets to the other one, so before the protein is able to fold, they form these big aggregates. That's a major problem in the cell, by the way. That's when you have all these ugly diseases like Alzheimer and Parkinson and many other diseases. Huntington, they come from the fact when the cell cannot control the folding and the proteins are able to aggregate, you have these diseases. So what Amson did on his experiment is he dilute these proteins so much so they didn't have enough time to come together. So in a very high dilution, he was able to observe the protein was able to self-assemble. In the cell, there's a mechanism to protect that protein so that it can self-assemble without aggregation. But in 1968, someone called Levinthal said there's a problem with this idea. And the idea is, how does this protein find this state? There's so many states to search for. How do you know it's the lowest energy states? And Levinthal created a particular problem that I'm going to tell you is the Levinthal paradox. It's written here in numbers, and it's good to play with this number. Let me give you an idea. Let's assume you have the first amino acid here from your chain. It's right on my hand. Here's the second one. Now let's imagine the second one, I'm looking for a particular shape. The second one can come on this orientation or on that orientation. Let's assume there are two possible orientations for each amino acid. If that's true, and you have two possible orientations for the amino acids, if you have 100 amino acids as a typical size of a protein, you have 2 to 100 possible choices. How many ways could you put that? Because you have 2 times 2 times 2 times 2 times 2, 100 times. If you could sample a state very, very fast, the fastest time you can think about it for anything is a picosecond. That's basically, if you really think, is 1,000th of 1 billionth of a second. 10 to the 12 seconds would take you 10 to the 18 seconds to fold. If you put this number equals to 5, if you bring this number to 1 billionth of a second, that's more reasonable. This just gets worse. But the bottom line is that proteins don't have this amount of time to search. So they cannot go and probe every structure and try to find out what's the lowest energy state. We wouldn't be alive if that was the case. So how the problem is solved? Experimentally, people said, oh, there's some memory in there. We never found that memory. What we came up is say there are other problems similar to this one. There's not a problem, like crystallization. So how proteins have solved that problem? This problem is not really a paradox. Why that's not a paradox? The problem when you have to search every state is the same idea than having a blind person on a flat golf course, trying to search where the hole is. So that's the time it takes. This time would take, the proteins have no guidance of where it was going. So what we need is some guidance. And that's pretty much what we are trying to represent on the next idea, what we call the principle of minimal frustration. So I'm going to try to put now one basic idea behind and of what sequence can fold. So here we come. If you had a random sequence of amino acids, and you figure out what's the lowest energy of these states. If you have a random sequence where you have A's and B's, A's and B's, A's and B's, what do you have? You try the B's, try to get together, the A's try to get together, but you never can satisfy all these interactions. So let's try to do this problem. Imagine you go to elementary school, and you get a classroom of elementary school, half boys and half girls. On this particular system, I can now do my problem. So now I'm going to have a heteropolymer, and the way I'm going to have a heteropolymer is, as a teacher, I have the power, and I say, boys and girls give hands, but I choose the order. A boy here, a girl here, a boy here, a girl here. I decide the line, and every child is giving a hand to other two childs. Now this is my sequence. It's a sequence of my A's and B's, but my A's and B's now are boys, girls, boys, girls, boys, boys, girls, girls, whatever I want the sequence. Half boys and half girls. Now what's the interaction? You know what happens at elementary school. At that age, you know what it is. Boys like boys, and girls like girls, and boys hate girls. It changed later in life. But at that age, that's what happens. So now you have this situation. No, so what the teacher says, get close to a friend. That's exactly what my problem is. My problem is like polars trying to get close to polar, non-polars trying to get close to non-polar. He is like boys trying to get close to boys, and girls trying to get close to girls. What happens, because of the chain, some kids end up a boy next to a girl. The one that's not fast enough end up to an interaction, there's a boy next to a girl, because they have the constraint of the chain. And that's exactly what we call a frustrated interaction. That's exactly what we call the concept of frustration. When they come together, you end up having a boy close to a girl. And if I repeat this experiment many, many times, you're going to have different shapes. And why you have different shapes, you end up with boys close to girls into a different place. In physics, that tells you that you have a frustrated system. And a frustrated system means that all these very low energy states that you're looking for, they're very different. Because although they're lower in energy, you haven't been able to make every interaction happy. You always have some interaction unhappy, because you cannot satisfy always boy, boys, and girl, girl. And the difference between this one, that's very different than this one, is because the pairs of unhappy boys and girls are different. So frustrated systems don't have a unique structure. Frustrated systems, you're going to have the structures you get, these low energy structures, they can be very different. That's exactly what you don't want into a protein. Proteins are designed to avoid that. So proteins try to put sequences that try for that particular shape to satisfy this interaction. So if I go back a couple of slides, you see what you try to do is, if you try to get a shape like that, you try to create a sequence where in the areas where you have the contacts, you always have the same color. So you try to design sequence where you have a sequence that matches the structure. And when you do that, instead of have many energy minimas that are very different, if you design it, you end up having one minimum dominate. So you want to pull one of these minimums down in a way that the stability of this minimum is large compared to the roughness of the landscape. In theory, we can now define very complicated things, means that you can come up and become stable at that structure. You can become stable at that structure before you get trapped on these minimums. So look what we have. What's the difference we solve this problem? Notice that if I look on the funnel idea, you have many, many states on high energy. Lots and lots of states. And you want to select one from these many states. In physics, when you have many states, what you call structural entropy, tells you you have many structures. So what I want to do is to have an energy bias, an energy drive that's able to balance this thing. There's enough energy, enough interest on coming down into that unique structure. So you can think about, again, the boys and girls. Basically, if you just have this line of boys and girls and they are moving around, they can have many, many different shapes. Why do they go to a unique structure? They go to a unique structure because they have this wish of getting a boy close to a boy, a girl close to a girl, and you try to satisfy that. And if you try to bias all these things, you have what we call a folding funnel. So let me try to continue into this idea that comes on this bias. So what you're seeing is a very simplified idea. So sequences that are able to avoid this frustration are going to try to solve this problem. Here what you have is a sequence on 3 different structures. A sequence where 1 is A, 2, 3, and 4 is B, 5 is A, 6 is B, and you have a sequence of 2 kinds of residues. And now to do an example, proteins are 3-dimensional objects. But just for visualization, I try to show you 3 possible structures. What we are doing in 2 dimensions, just as a cartoon. On the first one, you see the sequence has been designed for this particular shape in 2-dimensional. You see that every time you have dark green and light blue next to each other, they try to come together. You see here, together, together. Now, if I choose a different structure, the sequence doesn't like this structure. So for example, if I look at the second one, you can have a look around here, and you see these interactions are unhappy interactions. So for this particular structure, these interactions are what we call frustrated. There are more of them, like here. Here's just an example of frustrated interactions. So you see, this one is very well designed for this one. This one is not designed. If I go to the third structure, you see these also have frustrated interactions like you have down here. Right, so the bottom line now, you're trying to select. But this one has been optimally designed to avoid. There is one structure that satisfies all the interactions. So this is design, a direct map between the sequence and the structure. This very simplified idea has moved this field enormously forward. All I told you up to now is stuff that we were doing, believe it or not, 10 to 5 years ago. We are developing the framework, the funnel concept, the idea of minimal frustrations, or this base concept with these very simplified models. But these very simplified models now allow us to actually go and analyze real proteins. So I'm not going to come here and tell you about long simulations, but I'm going to try to show up a cartoon like that, where the same funnel I showed you before there, now draw on a more professional way, where now we call entropy. So let's see the base concept. That's what we call configuration entropy. That tells you we have many states up here, and we have just few states that are around the structure. Actually, don't have one state. You have a combination of states because the protein actually has to change its shape a little bit as its function. What important is the number here is very small compared to the number here. So you have many states. The question is, how do you select how do you beat this disorder? How can I choose one from many? I have to have enough energy drive, that's called the energy gap, that bring this state down. What I showed you before was a simplified model that showed you how the proteins come together. What we can do now, using the basic idea and try to design, I show here the example for one protein we call SH3. So what I'm picking up here to show this cartoon is a real representation of a very large simulation where you design these proteins, the concept of trying to minimize frustration or trying to make sure that your sequence is not frustrated, that you get the same idea. Up here, when you have many states, are the unfolded states. So when you have this black line tells you that you may have this particular necklace in complete disordered form. I show you 3 of them. As this thing is moving down the landscape, you start to search for parts to come together. But as you can see, in this particular case, some parts come early and then the other parts come afterwards. But which part comes, they're not necessarily the same ones all the time. And as they come down in energy, now you have states where you're satisfying more and more interactions. They're here. And in the end, you have what we call the folded ensemble. So what's the basic story here? What you see is that states that are very different are very high in energy. States that are very similar are very low in energy. And now what you have is, you start to form this structure. And how you form it doesn't make too much difference. So how you have is many routes bringing you down. Many routes bring you down. So you have this convergence of structure. This energy drive makes you to select. As you come down, you can identify. And the beauty of it is that you can identify these parts coming early, these parts coming. All that can be measured experimentally. So what do you have now? What you have now is a situation where you have simple models, where I give you the overall mechanism. You bring the simple models and use them to do the interpretation of very detailed simulations, where then you put all the information of the protein, and these things can be compared to a lot of experimental data that's out there. So you have a full integration of theory and experiments explaining how important these things are. So now you can learn things like, for example, if I get this situation here, and I do a mutation, I replace this residue by someone wrong, what's the consequence? How much energy do I lose? Do I have a different minimum? All these things can be explained. This base concept, this base figure, they came from very simplified models with this idea. So what was the idea? The idea is, here I have a necklace of beads. But these beads can be of different colors, and you can define interactions between these colors. Now can I figure out how this necklace finds a unique structure? I do that on a very simplified model and understand which sequences are able to have a unique structure and which sequences are not able to. The ones that are able to are the ones that minimize frustration, where all the interactions are satisfied. So you design to have that sequence. That's exactly what the proteins do for the sequence they're able to fold. And now you can go the extra step, and the basic ideas can be connected. So you come from physics, where you have the basic principles, you go through the chemistry of how these interactions take place between hydrophobic and hydrophilic, and you try to understand how biological function works. Thank you.